2 minutes. Just 2 minutes.
Okay.
Just 2 minutes.
Very good evening, my dear friends. Today's our eminent resource person, Dr.D.Nagaraju Garu, Assistant Professor, Department of Botany, Government City College, Autonomous, Hyderabad. Sir has huge experience in the field of mycology, especially fungi. So that's why today onwards, our resource person, our beloved brother, very huge experience personality, Dr. D. Nagaraju sir is going to explain about the entire topic related to our fungi regarding of this CPGET 2024. So that's why we would like to invite Dr. D. Nagaraju Garu, Assistant Professor, Department of Botany, Government City College, Autonomous, Hyderabad. So, sir, please welcome and share your screen. Start your session, sir.
Well, today we are going to discuss the very important, the second most largest group of living biota, that is fungi. You know which one is the world largest living biota? Anybody is there in the class?
Yes, sir.
Yeah. Do you know which one is the largest? That means highest amount of the living organism is there in which group? Either in plants or animal, or which group it having the highest group of living organisms? Do you have any idea?
No idea, sir.
You know, arthropoda insects is highest. Insects, arthropoda is the highest number of organisms, living organism in the world. After that, the fungi is the second largest group of living organisms. Fungi is the second largest group of living organism. So we are going to know a lot about the second largest group of living organisms that are surviving in our nature, that we are going to discuss. Okay, where the link actually in the certificate? Just a second, please. You know what are the fungi?
Sir,
What are the
sir?
fungi? Fungi means?
Fungi.
Fungi means?
Sir, it is fungi is nothing but bacteria.
Nothing but bacteria. Bacteria is nothing but fungi. Who told you? Fungi is nothing but bacteria. Though it is not. You don't say anywhere like that. Okay. Fungi differ from bacteria. Each and every organism having differentiation, their own structure, their own type characteristics. No two organisms are look like. Okay. There is variation is there. Lot of, the variation, evolution, variation, differentiation in all the living organisms. Darwin theory, Lamarck theory, you know. Do you know the Lamarck theory, Darwin theory?
Yes, sir.
What is the Lamarck theory?
The detailed I don't know, sir.
Okay, what does it mean? What does it mean? What it explains? What it explains? What does it? What is the Darwin theory?
Sir?
Darwin theory.
I'm not getting, sir.
Okay. Okay. You should know the theory. You know the Darwin theory is a theory of evolution, evolutionary theory. Darwin and Lamarck theories are two
Sir,
of
if
evolutionary
possible,
theories.
please click on slideshow, if possible.
Yeah.
Okay, sir.
Okay.
Please proceed.
Okay, now, the fungi is the second largest group of living biota. Just a second, I will see. Fungi is the second largest group of living biota which are there in the environment. These all are very useful in our daily life, which we are using in so many ways, in the so many things, either beneficially or harmfully. We are getting benefit or loss from the fungi. The study of fungi is called as study of
Sir.
study of fungi. Fungi, study of fungi, not pandi, fungi. Don't misspell the fungi. The fungus is a singular, fungi is a plural. The competitive exam is also asked which is the plural word of the fungi, which is the singular word of mycelium, either hypa or a hypa. The very, very important in the competition exam point of view, even in the daily life, we all should know about the fungi, what it is, how we are using, how the structurally, how we doing, how the nutrition is taking place, reproduction, and what are the basic structures. These all you should know. The basic and the largest group of plant biota among all the plant kingdom. As for a Whittaker classification, the Whittaker classification, Whittaker 5 kingdom classification.
Sir.
Whittaker 5 kingdom classification.
Yes, sir.
What it is?
Uh,
Whittaker classification. The living organisms are divided into 3. What are those?
Their class, order.
No. Total living organisms are divided into 5 groups as per Whittaker classification. What are those? I am asking. Monera, Protista, Fungi, Plantae, Animaliae. These are the 5 groups now.
Okay, sir.
Monera, Protista, Fungi, Plantae, Animaliae. Here, as per Whittaker classification, fungi are separated from the plant.
Okay, sir.
Fungi having the specific characters than the normal plant characters. That's why they separated, distinguished into different groups. That is as a fungi. Okay, it is the second largest group of living biota after the insect. The study of fungi is called as mycology. Study of fungi is called as mycology. Mycology, not my college, not yours college. Okay. Mycology.
Sir, what is that?
Study of the fungi is called as mycology. What it is? Not my college, not yours college. Okay, mycology. Here, myx means fungi. Myx means fungi.
Okay, sir.
Myx means, the molds and mushrooms. You might have seen the molds, so many areas where the spoiled food material, where on the bread, spoiled bread, roti, spoiled vegetables. You might have seen white mold, white booze, తెల్లని బూజులు చూస్తూ ఉంటుంటాం అవునా? You might have seen the molds. Even on the bread, roti, chapati, even vegetables, spoiled fruits, vegetables. Wherever it is mold, that is the fungi. And second most is the mushrooms. The mushroom? Mushrooms are the vegetable meats. We call it as vegetable meat. It having the more proteins and minerals than the meat. It is a very highly suggestible food even for the diabetic patients, old people also. It is very good nutrition diet, very good nutrition and diet for the diabetic patient. Here you see the molds, how it is upon the bread, how it's growing upon the bread. That is, the rhizopas, bread mold. We call it as bread mold or black mold or pin mold. The competitive exam, they ask, which one is the bread mold? Which among is the bread mold? Rhizopas, Mucor, Clostridium, Penicillium, yeast, like that they ask the question. The competitive exam point of view, we are going to discuss here, not in a theoretical class. Okay, we should go through each and every point in the question point of view. Each and every point in the question point of view, question answer point of view. Okay, that's the important for the preparation of the competitive exams. You don't go through the line by line as a theory, as a theory exam. Don't read out the para wise, page wise. Okay, where each and every line is very, very important while studying for the competitive exams. Okay, here you see the mics means hello, not hello mics. Mics means not hello mics. Mics means in the Greek, mics means molds or mushrooms. Molds and mushrooms. Study about the molds and mushrooms is called as mycology. Study about the molds and mushrooms. Study about the molds and mushroom is called as mycology. Okay, have you understood? What is the mycology? Study about the molds and mushroom. Nayana. Okay, study about the molds and mushrooms is called as mycology. The diseases which caused by the fungi is called as, the competitive exam they ask, diseases which caused by the fungi is called as mycosis. Mycosis. You might have listened in the corona time. Corona time you might have listened, mucormycosis, black fungus disease. Mucormycosis, black fungus disease. Have you heard? After recovering of the corona, from the corona, so many diabetic patients, they get infected with the black fungi, mucor. That fungi attack on the where the carbohydrate rich source is there. Usually in the sugar patients, it having the rich source of the carbohydrates in their blood. So it attacked and it caused death to the patients even after recovery from the corona. That is mucor or rhizopas. Mucor. Even actually it is a saprophyte. It decomposes the dead organic matter. It grown on dead plants and animals. It decompose and generate the minerals and nutrients. It supply the minerals and nutrition to the soil, earth, so that it cleans the environment. The most of the fungi are saprophytes. All the saprophytes are scavengers, natural scavengers. The means of scavengers, natural scavengers. Municipal, as like a municipal workers. Fungi and bacteria, it cleans the environment. Fungi and some of the bacteria, what are those are saprophytes that are cleans the environment, so that we could able to live with pleasant environment. Otherwise, if all the dead bodies, all the plant and animal dead organic matter will be remain upon the earth, you may not have no space. You don't have any space even to stand upon the earth. That is not happening because the fungi and some of the bacteria which are decomposing the dead organic matter, it is totally melting and get mineralized. Humidification, mineralization is taking place by these saprophytes. Get converting the proteins, lipids into minerals. That's the capacity of the fungi. That's why the fungi is called as, some of the bacteria and fungi, whatever the saprophytes, is called as natural scavengers. What are those? Natural scavengers. What are the toxins? Poisonous material, proteins which are there in the fungi. is called as mycotoxins. Mycotoxins. But how, listen, in the hostels and some of the areas, food poisoning. After heating the food, they got infected with the food. They are getting the dizziness, vomiting, stomach pain. That because of the contaminated food. They might have eaten the contaminated food that contaminated either by bacteria or fungi. You know the palli, groundnut seeds, wrinkled, small, minute size groundnut that occupy the fungi, and it decompose the total proteinaceous material, and it get converted minerals. While get converting the fungi, it also releasing toxins that are aflatoxins. You know the bacteria botulism disease, botulism, food poisoning disease, one of the food poisoning disease. The fungi, if it is causing by fungi, that is mycosis. One of the well-known fungal disease is mucormycosis, black fungus disease at the corona time. You know the chundru, dandruff. You might have seen the dandruff on most of the heads. That also caused by the fungi. Dandruff.
Green wave.
Yeah, dandruff also by microcystis fungi, that caused by the microcystis fungi, the very small minute fungi that cause the dandruff. Scalp will be eaten by the fungi so that it form the white granules like structure, that is dandruff, that caused by the fungi. Okay, you know what is the fungi? What does it mean? The definition of the fungi. The competitive exam they may ask. Fungi means, what is the definition? What does it mean, the fungi? Fungi means, fungi means, while studying this all, you should know the basic things. Okay, fungi definition is echlorophyllous heterotrophic thalopides. You know the classification, classification. Plants are classified into two groups. What are those two groups? You have an idea? Non-flowering plants and flowering plants. Plants are divided into two groups, that are non-flowering plants and flowering plants. Cryptogams and phanerogams. Cryptogams and phanerogams. Cryptogams, that means the non-flowering plant. They don't having the flower, fruit, leaf, root, stem like structure. There is no specific organization of the plant. Organization structure is not there. Okay. Unorganized thallus like structure. There is no flower. Non-flowering plants. Non-flowering plants are called as cryptogams. What are those? Cryptogams are called as non-flowering plants. Okay. The non-flowering plants again divided into how many? Do you have an idea? Non-flowering plants are divided into non-flowering plants. Cryptogams are divided into how many?
Five.
Cryptogams are divided into
5.
what? 5? Oh, what are those? What are those? Can you say? What are those 5 groups? Cryptogams are divided into 3 groups. What are those? Cryptogams are divided into 3 groups, that are, what are those? Thalopides, Bryophytes, and what are those? Talophytes, bryophytes, and pteridophytes. You know what the talophyte means? Phytes means not dishum phytes, not phytes. Okay, it's not phytes. Phytes means plants. In the Greek, phyte means plant. Plants which are having the thallus-like body. Thallus. That means flat structure, flatted structure. There is no root, stem, leaf-like organization. The thallus-like structure. Plants which are look like a thallus-like structure body are called as talophytes. Are called as talophytes. Okay. Thallus-like plant body which having are called as talophytes. Okay. Talophytes again divided into two groups. What are those? Do you have any idea? What are those two groups? What are those two groups? What are those two groups? Do you have any idea? Algae and algae and fungi. Algae and fungi. Two groups divided into two groups. First one is algae, second one is fungi. Algae means, what is it? Is the meaning of the algae? Definition of the algae. What are the algae? You should know. Even in the interviews, exams, they ask the question, what does it mean, the algae? What are the algae? Algae are chlorophyllous, autotrophic talophytes. Chlorophyllous, autotrophic, chlorophyllous, autotrophic. U is not responding. Huh? Okay. Chlorophyllus, autotrophic talophytes are called as algae. Study about the algae is called as? Study about the algae is called as? Huh? Study about the algae is called as? Algology, also called as Mycology. Also called as? Also called as?
Sir.
Study about the algae is called as? Mycology. Study about the algae is called as? Mycology. Okay. Phytes means algae, water moles. Okay. Water blooms. Phytes means algae. Logy means study. Study about the algae is called as Mycology. Not Phy college. Phy college kaadu. Kindi college, Phy college kaadu. Okay. I have understood. Not Phy college, not Kindi college. Mycology. Okay. Study about the algae is called as Mycology. Study about the fungi is called as Mycology. Study about the fungi is called as Mycology. Study about the fungi is called as Mycology. Study about the algae is called as Mycology. These two comes under, very important question. These two comes under, algae and fungi comes under
Talophytes.
Thalopides, yes. Thalopides are divided into two groups, algae and fungi. Okay, autotroph and heterotroph. Algae are autotrophs, fungi are
Heterotrophs.
heterotrophs. Heterotrophs means, what does it mean? Heterotrophs. Heterotrophs. Hetero, that means others. It depends upon others for their own food material. If they can't synthesize their own food material, it depends on others. It depends on host. It depends on other material for their own food material. Auto means self. They can synthesize their own food material, that are autotrophs. All the plants, almost algae or autotroph. Fungi and some bacteria are heterotrophs. Virus, bacteria, and fungi are heterotrophs. Not all the fungi, not all the bacteria are heterotrophs. Some of the bacteria are autotrophs also are there. You know, cyanobacteria, blue-green algae, cyanobacteria, that are autotrophic bacteria, photosynthetic bacteria. You know, these thalopides are divided into two. One is algae, second one is fungi. Okay, cryptogams are divided into three: thalopides, bryophites, and pteridophites. Thalopides divided into algae and fungi. Algae means autotrophic, chlorophyllous autotrophic thalopides. Plants which having thallus-like body and chlorophyll, it can synthesize their own food material, or algae. Plant like thallus-like structure, there is no chlorophyll and depend on others for their own food material, that are fungi. Okay. Okay. Study about the fungi, mycology. What happened?
So, it is fungi or fungi?
What it is?
Fungi or fungi, sir?
Fungi.
Okay, sir.
Fungi, not fungi. Okay, fungi. Don't misspell. Don't misspell. Take care while spelling. Spell out. Okay. And you know, the fungi, you know, the fungi are eukaryotic or prokaryotic? That also very important question. Fungi are prokaryotes or eukaryotes?
Eukaryotes.
Yes, eukaryotes. Fungi are eukaryotes. That you should remember. Fungi are not prokaryotes. The fungi are eukaryotes. Okay. Echlorophyllous eukaryotic group. Okay. And you know, who is the father of mycology? Who is the father of mycology?
Sir,
Who is the father of mycology?
Edwin John Butler.
Butler. Butler is our Indian mycologist, not the worldwide father of mycology. Butler is our Indian mycologist. Edwin John Butler is our father of Indian mycology, not the old mycology, not the world mycology, world mycology. Okay.
Okay, sir.
Myceli is the father of mycology. Okay. Mycology, Myceli, easy. Okay. He has written the Nova Plantarum Genera book. He mentioned about the fungi in that book. That also very important. Myceli written the book is Nova Plantarum Genera. What is the book it is? Nova Plantarum Genera. Nova Plantarum Genera book is written by whom? Written by whom?
Hi, sir.
Here it is there. You read out just so that you can remember. Mycology. Written by Mycology. Okay. Father of mycology. Okay. You know who is the father of modern mycology? Who is the father of modern mycology is Anton de Bary. H.A. de Bary is a German scientist. H.A. de Bary is a German. Father of mycology. Mycology is Italian. Okay. Anton de Bary is the father of modern mycology. Also, father of plant pathology, diseases, plant diseases caused by fungi, bacteria, virus, whatever it is. Plant pathology. Pathos means disease, logy means study, plant. Plant diseases, study about the plant diseases is called as phytopathology. Study about the plant diseases is called as phytopathology. Who is the father of phytopathology? Who is the father of phytopathology or father of modern mycology?
Anton D. Bary.
Huh? Who is
Anton.
that? Anton de Bary. Anton de Bary is the father of modern mycology. Also called as father of plant pathology, phytopathology. Okay. What you told, who is the father of Indian mycology? E.J. Butler. E.J. Butler is the father of Indian mycology. He studied elaborately, studied broadly about the fungi in our India. That's why he is the father of Indian mycology. Okay.
Okay, sir.
Our Indian mycologist, famous Indian mycologist is the Butler, and also Indian plant pathologist. Father of Indian plant pathology is who? Butler. Okay. You know the plant body of the fungi, we call it as thallus. Even the algae plant body also we call it as thallus. These two plant bodies we call it as thallus. That's why we grouped it as thallophytes. Thallus-like plant bodies are thallophytes. Okay, that's why the competitive exam they ask plant body of the fungi is called as thallus, tria, planta, thallus. Okay. Okay. And you see what it is. You see, there it is. Okay. You see what is the basic structure, fundamental structure of the fungi? They may ask the question. What is the basic fundamental structure of the fungi? Hypha. What it is?
Hypha.
Hypha. H-I-P-H-A. Hypha. Hypha is the basic structural unit of fungi. The basic structural unit. It's look like a tube-like structure. Tube. Vessels are tube-like structure. Filamentous-like structure. Tube or filamentous-like structure is called as hypha. Plural is hyphae. Plural word is hyphae. What is the singular word of the fungal structural unit? Hypha. Plural word of the fungal structural unit is hyphae. Okay, that you should remember. Hypha, hyphae. The competitive exam they will ask, what is the structural unit of the fungi? What is the structural unit of fungi? What is the structural unit of fungi? Hypha. Structural unit of fungi, hypha. Okay. You know what does it mean? The mycelium. Mycelium is the hyphal network. Hyphae which are interconnected and it forms a net-like structure. You see here, if the all hyphae are interconnected, there's so many hyphae. You see hyphae, number of hyphae, these are interconnected. The interconnected or interwoven hyphae are called as mycelium. Interconnected or interwoven or network-like structure, network of hyphae is called as mycelium. Complete example, mycelium. Mycelium is a network structure of hyphae. Or it is an interconnected hyphal structure is called as mycelium. Study about the mycelium is called as mycology. Mycelium. Okay, mycelium is called as mycology. You know, basically fungal plant body structure are two types. That is single cellular, unicellular, and hyphal structure, filamentous structure. Two types of the structures are there. What are those? Single cellular structure and filamentous structure, tubular structure. Two types. The single cellular structure, which having all the plant organs within the single cell. At the time of reproduction, all the cell will become a reproductive organ. It synthesizes the spores. That's why we call the single cellular fungi as called as holocarpic fungi. What does it mean? Holocarpic. Holo. This means all the plant body, fungus body will become the fruit. All the fungus body will become the fruit. That's why we call it as holocarpic. Holo means all, carpic means fruit. Okay, all the fungus plant body come into fruiting body, that we call it as holocarpic. You see yeast, chytridium, elphidium. Yeast, chytridium, and elphidium. These are the single cellular structures, single cellular fungi. You know the importance of the yeast? Not east, west, east. Okay, we have a lot of importance to the yeast also. Lot of importance to the yeast, sun rays. But even yeast, why yeast? Lot of importance is there to the yeast. If there is no yeast, no bakery, no beverages. No bread, cake, pizza, no bakery items. If there is no yeast, okay. If there is no yeast, no bakery items. No bread, cake, puff. These all being prepared by the yeast. What is the reaction of it is? Fermentation reaction. Fermentation reaction. Anaerobic respiration. You know the respiration types. Aerobic and anaerobic. Aerobic respiration means in presence of oxygen. It splits carbohydrates, glucose, totally splits into carbon dioxide and water. That is aerobic respiration. Anaerobic, that means in the absence of oxygen. That we also call as fermentation. That with that fermentation reaction, we could able to produce the bakery item and beverages, alcohol, drinks, beer, brand, whiskey, wine, vodka. all the items because of the single cellular yeast. Because of the single cellular yeast. $1 trillion of the money we are earning. So many are getting the food. So many are getting the drinks. To drink, to eat. Because of the yeast. If there is no yeast, no bakery items, no beverages item. No brandy shop, no wine shop, no bakery. That is the nature of this fungi, that is single cellular fungi, that is yeast, also called as Saccharomyces cerevisiae. Scientific name is yeast. Scientific name is Saccharomyces cerevisiae. That means myces means fungi. Shakara means shakara, sugar. Myces cerevisiae, fungi which is surviving on sugar. Fungi which is surviving on sugar. That's why I named it as Saccharomyces cerevisiae. Saccharomyces cerevisiae. Saccharomyces cerevisiae. Saccharomyces cerevisiae survive on sugar. That's why I named it as Saccharomyces cerevisiae. It is fermenting the glucose, converting into carbon dioxide and alcohol. Due to that, we could able to produce the bakery items and beverages. That is the greatness of the yeast, fungi. If there is no that fungi, what happened? If there is no yeast, what happened? No bakery, no beverages.
Bakery items. There is no bakery items.
Yeah, no bakery items we can't, no beverages items. If there is no yeast fungi. Okay. And you know one another most important fungi, penicillium. Alexander Fleming, penicillin. Teeth injection. Whenever we get injured, accident, that time we take the teeth injection, penicillin injection. As an antiseptic. That discovered by Alexander Fleming from the Penicillium notatum, Penicillium chrysogenum fungi. Penicillium notatum and Penicillium chrysogenus fungi that even nowadays, or even after 100 years, we are using the same penicillium whenever we get injured as an antiseptic injection. That is the greatness of the penicillium. No, the penicillium. Penicillium, pencillae. It looks like a pencil brush. It looks like a brush. You see here. That's why I named it as penicillium. Pencillae, pencil, look like a brush. Pencillae means brush. Look like a brush, paint brush. That's why penicillium. Okay. That is an. With that we are getting the penicillin. Penicillin, graciopulvin, this all are we are getting because of penicillium fungi. Okay, there is a lot of importance is there, the fungi. And then what is the basic structural unit of fungi is hypha. If a hypha network are interconnected, that we call it as mycelium. And the mycelium basically based upon the septa, various types are there. Septate hyphae. You see the septate hyphae, which having the septa. That means cross walls. Cross walls. The cross walls are there, that we call it as septate. If there is no cross wall, that is aseptate. No cross wall is there here, you see. No cross wall. There is no cross walls. Cross walls are absent. All the hyphae is the singular. That means all the nucleus are present within the single hyphae. That's why we called it as the cenocytic. What does it mean? if there is no septa in the hyphae, it having the multi-nucleus, that type of the hyphae, we called it as cenocytic. In the competitive exam, they ask cenocytic hyphae is present in Mycomycotina, Zygomycotina, Bacillomycotina, Asst.Mycotina. In which group of fungi the cenocytic hyphae is present? Which group of fungi? Which group of fungi? Zygomycotina fungi. Muca, Rhizopas. Okay. Pythium, Badaptera, Mucus, Rhizopas. In all this, there is no septa, cross septa. That's why that hyphae we calling it as cenocytic. Cenocytic hyphae, cenocytic thallus. That means multi-nucleus hyphae. Cenocytic means multi-nucleus. Multi-nucleus hyphae is present in Zygomycetes fungi, Muca, Rhizopas. Okay, the Muca, Rhizopas, multi-nuclear hyphae is present. Okay, the first one is septate, second one is aseptate, and the third one is, where it is the third one? The third one is simple porous septa and doliporous septa. Simple porous septa and doliporous septa. These are the two type of the hyphae. Okay, here you can see. Here you can see the first one is septate. First one is septate hyphae, that is there in all the higher fungi. Second one is aseptate fungi, that means there is no septa. That's why more nucleus are present in the single thallus, single hyphae. That is cenocytic, aseptate. Okay, the third one is simple porous septa. Septa, it having the small porous space. Septa fully not covered, fully not closed. The gap in between the two cells. That we call it as simple porous septa. You see empty space. Septa is not covered fully. That we call as simple porous septa. The competitive exam, they ask the question. Simple porous septa is present in Asst.Mycetes fungi. Which group of fungi? Asst.Mycetes fungi. Simple porous septa is present in Asst.Mycetes fungi. Aseptate hyphae is present in Zygomycotinous fungi. Zygomycetes fungi. Okay. Aseptate fungi is present in Zygomycetes. Septate fungi is present in almost all the remaining fungi. And in the septa, especially simple porous septa is present in Ascomycetes and doliporous septa. Dolipore, that means barrel shape, jar or barrel shape, or breaked coconut like structure. Breaked coconut like structure. This we call it as peranthosome. What we call it as peranthosome. The peranthosome and the porous space with the septa commonly called as dolipore septa. That means barrel shape or cylinder shape 4. You see the septa, it has swollen and it formed the barrel shape. From the barrel shape structure with the narrow space, that we call it as dolipore septa. Dolipore septas are present in Basidiomycetes. Which group of fungi? Basidiomycetes fungi. You should remember from this question, compulsory rights. Septate hyphae, accepted hyphae, simple porous hyphae, and doliporous septa hyphae. These are the four type of the hyphae. These are the four type of the hyphae which are having in the different group of fungi. These all four not there in the single group of fungi. These are different group of fungi which having various type of the hyphal structure. Okay. And then next we will go what is the cell wall component of the fungi? Cell wall is made up of it. The fungi cell wall is made up of it. Fungal cell wall is made up of it. Fungal cell wall is made up of it. Made up of it.
Sir. Sir, cell wall is made up of cellulose and pectin.
Cellulose and pectin. Who said? From where you know?
Sir, I read in degree book, botany book, sir.
Usually all the plant cell wall is made up of cellulose. But fungal cell wall is made up of chitin, not the cellulose. You should remember that is very, very important.
Okay, sir.
In the competitive exam, they asked fungal cell wall is made up of
Chitin.
Chitin. Somewhat variation, differentiation is there in each group of fungi from lower fungi to higher fungi. In some of the lower fungi, especially in the zygomystes fungi, that we also called as phycomystes. Phyco means algae. Fungi which are look like algae is called as phycomystes. Competitive exam lo adugutadu. Phycomystes. It looks like a phyx, algae, algal-like structure. That's why phycomystes. Okay, especially Oomystes, Catidomystes, that fungi is also called as phycomystes. In that, along with the chitin, cellulose also present. Okay, cellulose plus chitin. In the higher fungi, along with the chitin, glucan, mannans are present. In Ascomycotina, Basidiomycotina, along with the chitin, glucan and mannans are also present in the Ascomycotina and Basidiomycotina. In Ascomycotina and Basidiomycotina, along with the chitin, what are the ingredients which are there in the cell wall? Glucan and mannan. Glucan and mannans also present. Okay. You know what is the storage food material of the fungi? What is the storage food? What it is?
I don't know, sir. I'm saying.
You don't know. Okay, good. Fungi storage food material of fungi is glycogen, as like a lipids, not the starch. Majority of the plant storage food material is the starch. But fungal storage food material is glycogen. Fungi and bacteria storage food material is glycogen. In the bacteria, the PHB, poly-beta hydroxybutyric acid, also present, that we are using as a biopolymer. Okay, how the fungi will stain. In the experiments, you might have studied the section cutting of the diseases. white rust, wheat rust, Tika disease, that you have taken the section. What stain you have used? The fungal identification, fungal stain. What is the fungal stain? That also asks the question in the competitive exam. That is lactophenol cotton blue. What it is? Cotton blue. We use the cotton blue so that it will be the blue in color. It will be the blue. That's why with the blue stain we can easily identify the fungi. Otherwise, the fungal mycelium is in white in color, hyaline, no color. Some of them having the brown color, but mostly the hyaline in color, no color. To identify, to distinguish, we add the stain of lactophenol cotton blue. That is the fungal stain. Very, very important in the exam, competitive exam. Fungal cell wall and fungal stain. Okay. Okay, some of the fungi, it shows the dimorphism. Dimorphism. That means it also can live as a saprophytes and also as well as a pathogens whenever the favorable condition is there. Whenever the favorable condition, it also acts as a parasite. It can change their structure. You know, the lung disease, histoplasmosis, lung infection is caused by histoplasmus capsulatum. That shows the dimorphism. Even yeast also shows the dimorphism. Okay? And, you know, the homokaryotic and heterokaryotic mycelium. Homo means identical, same, similar. Identical, similar structure, similar nucleus. The nucleus having the same identical, that are homokaryotic. Heterokaryotic means two different genetic nucleons. That means either one from mother, one from female, or one is plus or another one is minus. Heterokaryotic. You see, already told you multinucleate. Multinucleate, that is there in the cenocytic thallus, especially in the zygomystes. Okay. You know, one of the fungi, that is Microsporidia, which locks the mitochondria, peroxisomes, and centrioles. Usually fungi which locks the chloroplast. But even in the Microsporidia, mitochondria, peroxisomes, and centrioles also absent. Okay. Okay. Are you listening?
Yes, sir, I'm listening.
Okay, good. Then, you know, you see the chitin structure, how it is. And what is the difference between algae and fungi? Can you say any two different distinguishing characters? What is the difference between algae and fungi? You know, the algae are mostly aquatic, it live in water. Fungi are terrestrial, it live on earth, soil, or any host. You know, algae having the chloroplast. Chloroplast is present in algae.
Lives in only land, sir.
Which one? Fungi. Yeah, land means any host. Saprophytes which live on land, and the parasites which live on host, which cause the disease to the host. Pathogenic fungi.
Okay, sir.
You know, the trophic levels, that means nutrient level, nutrition type of the fungi, that we will discuss later, next. Okay, heterotrophs again divided into three groups: saprophytes, parasites, symbionts, and necrotrophic, four types. are there. Okay, that we will discuss. Saprophytic fungi are decomposed fungi. It cause no disease, no harm. It clean the environment. It as a decomposers. Okay, and then what is the difference between algae and fungi? Algae having the chlorophyll, fungi it absent. Okay, algae are autotrophic. They synthesize their own food material. Fungi are heterotrophic in nutrition. They depend on others as a saprophyte or a parasite or a symbiont or an ectotroph. Okay, almost all the algae are eukaryotic except cyanobacteria, blue-green algae. All the fungi are eukaryotic. No prokaryotic fungi. Okay. Okay, you know the cell reserve food material of the algae is starch. Reserve food material of the fungi is glycogen, oil glands. Okay. There is no parasitic members in algae, but parasites are common in the fungi. No dikaryotic phase, that means two nucleus stages absent in the algae, but two nucleus stages commonly present in the higher fungi, Ascomycetes and Basidiomycetes fungi. Okay, and what are the other difference? These are the main distinguishing differences between the algae are aquatic, fungi are terrestrial. Algae having a chlorophyll, fungi are chlorophyll, no chlorophyll. Algae are autotroph, fungi are heterotroph. Okay, algal cell wall is made up with cellulose, fungal cell wall is made up with chitin. Algae storage food material is starch, fungal storage food material is glycogen. These 6 differences is enough that you should remember. Okay. Where it is, what happened? Why is not? Okay, then we will go for the presentation. That's good. Then that. Okay, well, you know the 3 types are the fungi, fungal structures, fungal states. What are those? Telomorph, anamorph, and holomorph. From this also question raised. You know the first aspergillus kept in the Deuteromycotina group, fungi imperfecta group, dustbin fungal group. You know, Deuteromycotina fungi also called as dustbin fungi. Deuteromycosis fungi also called as dustbin fungi are also called as imperfect fungi. No perfect stage. No perfect stage. Perfect stage is absent. Perfect stage is absent in the Deuteromycotina. That's why we call it imperfect fungi. First, whatever the fungi we identified that kept in the Deuteromycotina group. After identification of the sexual reproduction stage, fruiting body, then they shift into other groups, either Masticomycotina, Zygomycotina, Ascomycotina, or Basidiomycotina. Okay, here you see first asexual stage of the fungi that we called as anamorph. Asexual stage of fungi, what we call anamorph. Okay, anamorph. And whenever we identify the sexual stages, then that shifted to other perfect group of fungi. That perfect group of fungi, whenever we shifted into that we call it as telomorph. That means sexual stages. Perfect stage of the fungi is called as telomorph. Both combinedly we call it as telomorph and anamorph is combinedly called as holomorph. Combinedly called as holomorph. This is very important. You know, the Aspergillus, after identification of the perfect stage, they changed the name it as Emersella nidulans. Emersella nidulans. Instead of Aspergillus nidulans, given the name of Emersella nidulans. As like Penicillium also. Penicillium is the imperfect stage. And then shifted into Ascomycotina after identification of the fruiting body. Like that, having the imperfect and perfect stages. Imperfect stage is called as anamorph. Perfect stage is called as telomorph. Okay. You see the difference between homothallic and heterothallic fungi. That means the two thallus, that means male and female nuclei both are from the same thallus, that is homothallic. Male and female, that means plus and minus nucleus, if they are in the same nucleus, same thallus, that we call it as homothallic. But if the male and female are separate, that means plus 10 thallus and minus 10 thallus, if it is separate, that we call it as heterothallic fungi. You know the heterothallism? Heterothallism. Heterothallism. A. Blacksley first identified the heterothallism. Heterothallism. That means two different thallus. In between the two different thallus only it do the reproduction, sexual reproduction. It takes the sexual reproduction. It is not happen in the same thallus. The same thallus, it having the only one type of the either plus or minus strain of the nucleus is present, not both. If the both are present, that is homothallic fungi. If the plus 10 thallus, plus 10 nuclei is present in one thallus and minus 10 nucleus is present in another thallus, that means in the different thallus, that we call it as heterothallic. That means if the same thallus to hyphae, it come together, they can't mate. Because of it having the same type of the nucleus, same strain, either plus or minus. If it is from the different thallus, that may be the minus strain, then it can mate in between the plus and minus strain. That is called as heterothallism. Heterothallism, that means self-sterility. If the thallus of the two hyphae of the same thallus will not fuse, will not take the fertilization. Between the two opposite strain, thallus only take the fertilization. That is called as heterothallism. That first identified by A.F. Blacksley. Who is that? A.F. Blacksley. Blacksley first identified. Blacksley first identified this heterothallism. Heterothallism is very important. Heterothallism is present in Mucor, Bisido, and some of the Aspergillus species and Mucor species. Heterothallic fungi are present. Which among is heterothallic fungi? They may ask the question. Mucor, Bisido, and Aspergillus nidulans, like that fungi, which shows heterothallism. It can't mate between the same thallus hyphae. It need to opposite strain thallus to mate, to do the reproduction, fertilization, to take the fertilization. That is called as heterothallism. First discovered by A. Blacksley in Mucormycido. First identified. Heterothallism identified in Mucormycido, identified by A. Blacksley. Okay. And then, I already told you two type of the fungal thallus, unicellular and hyperstructure, microscopic and macroscopic. Macroscopic are mushrooms, the mushrooms. And first one is unicellular, second one is filamentous. Filamentous fungi which having the hypa, hypae, I already told you, hypae, interconnect hypae called as mycelium. Okay. And then you can see here the septate mycelium and aseptate mycelium. Aseptate mycelium having the multinucleus, that means cenocytic. You see the fungal stains, fungal on the fetides, fungal cultures. You see, I already told you the dimorphism. Dimorph fungi, it shows the dimorphism whenever the temperature is increased above 36, 37 degrees centigrade. Okay, you see some of the examples of dimorphic fungi: Histoplasma, Blastomyces, Coccidioides, Paracoccidioides, and Penicillium, and Sporothrax. These all are so the dimorphism due different structure. And already told you the cell wall, fungal cell wall is made up of it, chitin, but in some of them, along with the chitin, what are they? Glucons are present in oomycetes, that means in the one class of Zygomycotina. And the chitodomycetes, chitin and glucan is present. The Zygomycetes, chitin and chitosomes. Chitin and chitosomes are present. Chitin and chitosomes in the Zygomycetes fungi. You see, chitin and glucan in the Chitomycetes fungi, and Ascomycetes and Basidomycetes fungi. Along with the chitin, glucan also present. And especially in the Saccharomyces, Cryptococcus, glucan and mannan, some of the Ascomycetes fungi. Mannan and chitin in some of the Basidomycetes fungi. There is a variation for that. But majority is the chitin. But in the Phycomycetes, along with the chitin, cellulose also present. That you should remember in the cell wall structures. Okay. The fungi having the specific structure that we call it as hostorium. Hostorias. Fungi with having the specific structure that we call it as hostoria. Hostoria means it is a finger-like projection or vesicles or finger-like projection which penetrate into the host tissue. Especially these all are present in parasitic fungi, not in saprophytic fungi. Not in saprophytic fungi, it are present in the parasitic fungi. Parasitic fungi which having the adaptation, specific structure to absorb the nutrition from the host. You see it having the specific vesicle-like structure which are penetrate inside the host. This is the host cell, not the fungal cell, host cell, either any plant or animal, whatever it is. The fungal mycelium which are penetrating into the host cell. That specific structure is called as hostoria. There is a 5 types of the hostorias are there. Finger-like projection or vesicle-like and intracellular and intercellular and multi-branched. See the finger-like projection mycelium and vesicular shape mycelium and branched type of the mycelium and intracellular, that means hyphae which are enter into the cells, and intracellular, that means in between the cells. In between the cells, the hyphae is penetrate, form the network. That is intracellular and intercellular, vesicular, and finger-like projections and arbuscular. That may, and then the multi-branch. These are the various type of the Hasturias. Shall I stop? It is already 1 hour, near to 1.5 hours. In the next class, we will discuss the nutrition. Okay, either nutrition or classification or reproduction. Reproduction of fungi is there, nutrition of fungi, and classification of fungi, economic importance of the fungi. These all are we are going to discuss, very, very important. Okay, all the type studies will cover in this. You need not to go each and every type study. Yeast, Penicillium, Puccinia, Albugo. This all will cover in the general characters. What we have studied at the thallus organization, thallus structure, general character, cell wall, cell structure, and hyphae types, mycology, and various stages of the fungal states, telomorph, anomorph, holomorph, and various type of the hyphae, aseptate, septate, simple porous septa, daldipore septa. Okay, and the difference between algae and fungi, and fungal basic characters, that all we have discussed. Okay, then we will discuss the nutrition tomorrow. Okay, we will discuss the nutrition tomorrow. 4 type of the nutritions, modes of nutrition in fungi, that we will discuss tomorrow. Okay. Okay, bye. Tirupati. Okay.
Yes, sir.
Okay, right. In the next class, we will discuss the nutrition, reproduction, life cycles, classification, economic importance. Okay, these are the topics are there. From the you will get the more questions in the even any competitive exams. You will get the more questions from the fungi. Okay, lot of questions. Till now we have discussed lot of questions, important question point of view. Study about the fungi, fungal structure, basic structure of fungi, mycelium, hypha, mycelial structure type, hyphal type structure, hyphal structure type. Okay, some basic importance, dimorphism. These all we have discussed. Okay.
Okay. Very good evening, my dear friends. Very, very important and the longest class in the entire CPGET 2024 class. Really, we are very happy and thankful to our respected resource person and my beloved brother, Dr.D.Nagaraju Garu, Assistant Professor, Department of Botany, Government City College, Autonomous, Hyderabad. Really wonderful session, my dear friends. Don't forget and listen carefully. I will send the YouTube video also. It is very, very, each and every point is important. Not only in fungi, not only in microbiology. It is very related to the basic information related to the Thallophyta, including pathology, plant pathology, And also most important points regarding the father of microbial, plant pathology, father of fungi. These are the most important questions related to the each and every point of today's class is most important. So thank you very much, my dear beloved brother, Dr. D. Nagaraju garu. Tomorrow, same time we will meet.
Okay, right.
Hats off to you.
Okay, right.
Excellent
Bye,
class.
bye, bye, bye.
Excellent
Bye,
class.
bye, 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 bye.
Thank you.
Right, right.